Now, this last part of Chapter 4 will be discussing neurotransmitters and their actions. Neurotransmitters are chemicals that are made by the nerves and released from the axon terminal, and they lead to certain changes to either a nerve or an effector. There are at least about 100 neurotransmitters. We are not obviously going to learn all of them. We're not going to even learn most of them. We're going to learn some of them, okay? Just, ones that are of extreme importance. For example, acetylcholine. Acetylcholine, that is the neurotransmitter of the neuromuscular junction. So how a nerve transmits the impulse from a nerve to a skeletal muscle is thanks to the neurotransmitter known as acetylcholine. But neurotransmitters can be made out of amino acids. They could be made out of monoamines. They could be peptides, so a short peptide chain. They're just different kinds of neurotransmitters. They are produced in the rough ER or in the cytoplasm, and they would have to be transferred along the axon and stored at the axon terminal in those little synaptic knobs. They are stored in a vesicle and then released via a process of exocytosis. Now, these are the neurotransmitters that we want to focus on. So, for example, acetylcholine. As I said, it is one of the most important neurotransmitters in the body. It is the neurotransmitter of the neuromuscular junction. So this is how the nerve tells the skeletal muscle to contract. So it is found in the CNS at the neuromuscular junction. Norepinephrine, also known as noradrenaline. In the CNS, it creates a sense of well-being. And that's why low levels of norepinephrine can lead to depression. Dopamine is another neurotransmitter in the CNS, and that creates a sense of well-being. Deficiency of dopamine has been implicated or associated with the development of Parkinson's disease. Serotonin is a neurotransmitter found in the CNS. It is an inhibitory neurotransmitter, so serotonin would actually lead to sleepiness. And we will see that serotonin has also been implicated in depression. Okay, you could see kind of like that relationship between depression and sleep disturbances. Serotonin's action is blocked by LSD or acid. And one of the ways that we could, treat depression would be through drugs known as SSRIs, or selective serotonin reuptake inhibitors. That would increase the levels of serotonin in the CNS, and in an attempt to treat depressive symptoms. Histamine is found in the CNS as well. It is released in the hypothalamus and it promotes alertness. So histamine keeps us alert, and that's why a lot of the antihistamines out there that are allergy medications, they can lead to sleepiness because they are antihistaminics. They decrease the level of histamine. Now we will move on to these amino acids. We've got GABA and glycine, and these are inhibitory, while glutamic acid or glutamate is the excitatory neurotransmitter. Both of them are found in the CNS. For neuropeptides, in the CNS we have enkephalins and endorphins, and these are inhibitory, meaning that they would inhibit the neurons. They reduce pain by decreasing another neurotransmitter known as substance P. So substance P comes from pain, is released in the peripheral nervous system. It is excitatory and it leads to pain perception. It increases our sense of pain. While enkephalins and endorphins balance that by decreasing the amount of substance P being made. And that is why, there is a relationship between the levels of endorphin and pain perception. Endorphin is also another neurotransmitter that is associated with depression. It's also associated with premenstrual syndrome. So the pain, the symptoms that some women develop right before their menstruation. That would be due to the low levels of endorphins, and that leads to a higher pain perception. Now, where can we find endorphins naturally at the drugstore? You could, or in any market. Actually, dark chocolate is full or is rich with endorphins, and that is why some women before their cycle have that urge or need to eat chocolate. It's a sort of self-medication where they are intaking endorphins, and that elevates the mood, and it also decreases their perception of pain. Some gases act as neurotransmitter. For example, nitric oxide in the peripheral nervous system is a vasodilator. It dilates blood vessels. So, for example, the blood vessels in the penis during an erection, nitric oxide will be released from the neurons, leading to the vasodilation, causing that erection to occur. Now, these are some disorders that are associated with neurotransmitter imbalances. For example, we've got clinical depression, and unfortunately, depression does not have one cause, and it's not like one cause fits all kind of thing. There's lots of different reasons. Everybody's very different, and that's why one medication that works for one patient might not work for another. So it could be due to the deficiency of norepinephrine, deficiency of serotonin. It has also been implicated deficient of dopamine. So there's just so many different reasons why, chemical reasons of depression. Hypersomnia and insomnia. So hypersomnia is excessive sleeping. Insomnia is the inability to sleep. They are both related to serotonin. Okay, so excess serotonin would lead to excessive sleeping, while deficiency of serotonin would lead to the inability to sleep. And again, you can see how serotonin levels leads to sleep disturbances, and it can also lead to depression. And you can see how that is true when we think about what a depressed patient would have. They would also have sleeping disturbances. Now, mania is a patient that has too much norepinephrine, and that would lead to elation, irritability, over-talkiness. So they tend to talk, for a very long periods of time. And they have these increased movements. They move a lot. They talk a lot. They could stay up throughout the night, vacuuming or doing laundry or so on. So that is due to the excessive norepinephrine levels. Parkinsonism is where a patient does not have enough dopamine, so due to the deficiency of dopamine. And you want to think of Muhammad Ali. He had Parkinsonism, and you could see the tremors that he had, the slow movements and the muscle rigidity were very clear. Another patient another actor is Michael J. Fox, who also has that same disease, and you could see these manifestations very clearly. Schizophrenia is due to the deficiency of GABA, and that would lead to excess dopamine. So schizophrenic patients have inappropriate emotional responses to different stimuli and visual or auditory hallucinations. So what are some drugs that alter neurotransmitter levels? There's a lot of them. Okay, we're going to tend to focus on the ones that are more common within the community. So for example, nicotine. Okay, these activate cholinergic receptors. Cholinergic receptors are the receptors that would usually be activated by acetylcholine. So nicotine are going to activate those same receptors, and that leads to the increase in alertness. Remember that acetylcholine is also the neurotransmitter of the neuromuscular junction, and that's why after cigarette smoking you get the jitters, because of the stimulation of those same receptors. Tricyclic antidepressants, MAO inhibitors or monoamine oxidase inhibitors, and SSRIs. Okay, all of these can be used as different options to treat depression. So the tricyclic antidepressants, they increase the levels of norepinephrine and serotonin. MAO inhibitors increase levels of norepinephrine. while SSRIs increase the levels of serotonin. And again, you can see that, what works for one patient might not work for another, and it's all about trial and error. So what are the events that lead to the release of the neurotransmitter? And I want to go to an image because we already talked about this, but let's go over it one more time. So bear with me here for a minute. There we go. So what leads to right here again is the end of the axon. So your axon terminal with a vesicle, and the neurotransmitter is inside of it. It's stored in that secretory vesicle. What are the events that lead to the release of the neurotransmitter? So remember, when the nerve impulse comes to the end of the neuron, opens up calcium ion channels, calcium rushes into the neuron, and that leads or assists in the release of the neurotransmitter through exocytosis. So what happens to the wall of the vesicle? It blends or becomes now part of the plasma membrane of that neuron. When things become endocytosed, we can reuse that, we'll regain that membrane back into the secretory vesicle, and that is known as the trafficking of these membranes. So we don't really lose the membrane. So bear with me one more time here, folks. Now we are going back to the events. So when the action potential moves along the axon, it reaches the synaptic knob. That opens up your calcium ions channels, and calcium goes inside of the neuron. That would help the synaptic vesicles to fuse to the synaptic knob, releasing the neurotransmitter by exocytosis into the synaptic cleft. Now the synaptic vesicle membrane becomes part of the cell membrane. And remember we said that we could regain them through endocytosis. That is known as vesicle trafficking, where we are basically recycling the membrane. It goes from the membrane of the secretory vacuole to cell membrane and keeps on going back and forth. A neuropeptide is a kind of neurotransmitter that is made out of a small protein chain. We already talked about these: enkephalins, endorphins, and substance P. I just wanted to kind of touch on the difference between a neurotransmitter and a neuromodulator for a second here. So neurotransmitters are the ones that we're talking about. So these transmit information or transmit the action potential from one neuron to whatever's after that nerve. While the neuromodulator modulates the response of the postsynaptic neuron or the effector to the neurotransmitter. So your neurotransmitter is the main actor here. The neuromodulator can sometimes modulate the response a little bit. Neurotransmitters produce an effect faster than neuromodulators. Neuromodulators are more also important in long-term things. So for example, learning process. So neurotransmitter, you are hearing me, you're seeing me. These are all things that go through neurotransmitters. They're instantaneous stimuli, while the neuromodulators in the brain are trying to process all the information and tying it into other things that you've learned, ingraining it into your neurons. That process takes a little bit of a longer time, and those would be mediated through neuromodulators. So a little clinical application here, which is really important, would be the effect of opiates in the human body. So we talked about endorphins. Remember, those are actually our internal or our endogenous opiates. So we make our own opium, so to speak, and morphine and heroin, and those are your endorphins. But opiate drugs, external sources like morphine, heroin, codeine, and opium, they are very potent painkillers that are made out of poppy plants. And they activate the exact same receptors as these endorphins do. The thing is that if somebody takes them over and over again, that will inhibit that person from making their own endorphins. And that would lead to severe withdrawal symptoms if these opiate drugs are stopped suddenly. So the opiate drugs are very useful to relieve severe pain, but they're also very addictive. And again, that is due to the fact that they would experience severe withdrawal symptoms, especially pain, when they stop taking these drugs, because again, due to the long exposure over time, now their brain is no longer making their own endogenous opiates, and it needs to be gradually withdrawn from the body to give the chance to your CNS to start making their own opioids just like they used to. This is a little story that I actually read in the paper. I thought it was really interesting, where it's an unfortunate event where the mom ate a poppy seed bagel and then didn't pass a drug test. Okay, and then I want you to guys kind of watch and see what happened in this. It does have a happy ending, so that's a good thing. Next thing would be the impulse processing. So how is it that nerves process all of this information? Okay, the different things are coming at it. Some things are excitatory, some things are inhibitory. Now, how is it that the nerve, this poor little nerve, kind of decides what to do? There is a process known as facilitation. And facilitation would be where, if the nerve keeps on being stimulated over and over and over and over and over again, at that point, it is going to be releasing a lot of neurotransmitter than it would usually do if it was just one impulse. Okay, that is facilitation. And that obviously will increase the likelihood that that postsynaptic cell will reach threshold levels and become excited. Now we have these different neuronal tools, and it kind of works like groups. So kind of like how we in class, we divide the students kind of into groups. So one group for discussions, other groups for group presentations, and so on. Nerves do the same thing. So nerves kind of gather together where, okay, this group of nerves are going to be for memories. This group, other group will be for maybe smell, and so on. So these are known as neuronal groups, where you have a group of neurons that synapse together and they have kind of like one common function. And we are talking here in the CNS. Okay, so these are inner neurons that we're talking about. They work together to perform a common function. They do not necessarily have to be in the same part in the brain. They could be pretty much scattered all over the brain, but again, they're all connected together. And these pools will definitely have kind of like extensions to the outside. So they will be connected through the sensory and to the motor neurons in order to gather information, process it through these inner neurons, and then send out motor impulses to the effectors. And you can see how this works. So when I go through this example, you'll get a better idea of what we mean by these neural pools, and then convergence and divergence. So convergence means that lots of information is converging on or is being funneled into this one nerve. So one nerve right here, so this is nerve number, let's just call it nerve number 3, is getting input from these two nerves, nerve number 1 and number 2. So again, it's being funneled onto or converging onto this one nerve. And you want this in order to gather as much data as possible and to be able to analyze it. So think, for example, I'm crossing the street, okay, and I have one nerve that is seeing a car, and then I have another nerve that can hear a car. Okay, if these two pieces of information did not converge onto one nerve, there would be no way to put these two pieces together to understand that I'm about to get hit by a car, okay? And that is the importance of convergence. Convergence allows the brain to gather as much information as possible and integrate all of that info together in order to figure out what's going on. Okay, so it allows the nervous system to collect, process, and respond to information. Now you want to contrast that with divergence, where you see here this nerve number 4 sending out impulses to nerve 5 and nerve 6. Okay, so this leads to the amplification of an impulse. So going back to that example where, nerve number 3 knows now that I'm about to get hit by a car. Nerve number 4, there's also an interneuron, decides, well, you know what? We need to do a couple of things. First off, you want to start running. You might want to stimulate the heart to go faster. You might want to stimulate the breathing to go faster, again, to be able to run. So this one interneuron will send impulses to my skeletal muscle fibers through nerve number 5 to contract and hence to run. It'll send impulses to the heart through nerve number 6 to be faster. And then maybe through a third neuron number 7, it would send impulses to my lungs to breathe faster, to the diaphragm to breathe faster. Okay, so again, you have this process of amplification. You're able to spread the information to more than one resource or to more than one effector. Okay, and that again is known as the process of divergence. So this whole process of converging information and diverging information, this all kind of ties into the neuronal pool. Now going back to drug addiction, that generally speaking, drugs alter the neurotransmitter at the postsynaptic neuron. They either stop it or increase the synaptic transition. An agonist would be a drug that would activate the receptor in the postsynaptic neuron, while an antagonist would bind to the receptor and block it. Okay, so these are two different kinds of drugs. Both can obviously be harmful or could be useful if they're used in treatments. Okay, so the repeated use of an addictive drug causes a decrease in the receptors. So the cell has the ability to figure out, you know what, there's just way too much of, let's say, heroin coming into the body. I am going to remove some of my receptors. That is known as down regulation of the receptors. At that point, that means now that this person is going to have to take more heroin, so increase the dose, in order to get that same effect. Okay, that process of upping the dose because of the loss of the receptors is known as drug tolerance. Okay, so again, the repetitive use of the addictive drug causes a decrease in its receptors, so the person would have to take more of the drug to have that same effect. Okay. Amphetamines, these are just a couple of examples of commonly abused drugs. So amphetamines, that enhances norepinephrine activities, it elevates alertness and it elevates the mood. Nicotine, that is found in cigarettes, these stimulate the acetylcholine receptors. Those are known as nicotinic cholinergic receptors. And that leads to the pleasurable feeling from smoking because it increases the dopamine release. And remember that because acetylcholine is the neurotransmitter of the neuromuscular junction, it also leads to shakiness that could be seen after smoking too. So this ends chapter 10. Let me know if you folks have any questions.